Dear viewers, welcome to my video lecture. My topic name is Bacterial Endospores. What is mean by endospores? Endospores are produced by bacteria during an adverse environment by producing an heat, radiation, chemicals and etc. Endospores are the resisting non-reproductive structure formed by bacteria for the survival during adverse environmental condition. Endospores were first discovered by John Tyndall by producing a heat at the bacteria. Endospores are structurally, metabolically and functionally different from vegetative cells. Endospores are commonly found in soil and water. It may survive for long period of time in this environment. Now let's see about the structure of endospore. The structure of endospore is very complex and they possess multi-layer covering. The outermost layer of the spore is called as a exosporium, which is relatively thin and duplicate in nature. Beneath the exosporium, spore coat is present. Spore coat it contains serve a layer of proteins. It is thick in nature. Due to this thickness, high resistance of endospores towards heat, radiation and chemicals. It protects the bacteria. Inert to the spore coat is cortex. It is thicker wall layer in the endospore. Cortex is very large. It occupies the half of the spore volume. Cortex is composed of peptidoglycan. Peptidoglycan is a cortex less cross-linked than vegetative cell. Innermost layer of the spore is called as a spore coat. Spore cell wall cover the central protoplast. The endospore core has a normal cell structure as that the vegetative cells. The core contains ribosome and centrally placed nucleotide, genetic material DNA. This core contains 10 to 25% of water of the normal vegetative cells. This is the structure of endospores. Here we can see the parts of this endospore. Outer layer exosporium, outer coat, inner coat, cortex, diplococcal acid, ribosomes, DNA, small acid soluble binding protein, core containing cell genome, inner membrane and core wall. Now let's see about endospore producing bacteria. Endospore production is associated with gram-positive bacteria. The gram-positive bacteria only produce the endospore. The gram-negative bacteria does not produce endospores. Few genera of the bacteria produce endospores such as Clostridium species gangrene and Bacillus species anthrax. This both bacterial species are gram-positive rods shape. Some other genera are also produced endospores. They are Desulfatomaculum, Sporolactobacillus, Sporosarcina, Oxalospira and Thermoactinomycetes. This bacterial species also produce endospores. Let's see about the functions of endospores. Endospores are ultimately production for bacterial genome. Endospores is used for the production of bacteria. Spore form within the cell and contain full copy of bacterial genome. Endospore resistance against heat, boiling, drying, antibiotics, low nutrient condition, radiation, osmotic pressure, various chemical disinfectant. Now let's see about the sporulation of sporogenesis. This is the process of formation of endospore within the vegetative cells. This process takes place several hours only. This takes place in stationary phase of the growth curve. Now see this sporulation. DNA replicate extend into an axial filament. This filament become long and septum form near one pole, separating the four spore from mother cell, and each get chromosomes. After that, mother cell engulf the four spore and started to surrounding the four spore has a secondary membrane. Chromosomes of mother cell start to break down and four spore develop the cortex layer of peptidoglycan between the original four spore membrane or from the mother cells. The protein diplococcal acid is synthesized and calcium is incorporated into the spore coat, and then mother cell release the spores. Here you can see the vegetative growth. Vegetative cells are divided by binary fission, asexual reproduction by the separation of body into two new bodies. An organism duplicates its genetic material and divides into two parts, and each new organism receiving a one copy of the DNA. This is the sporulation of a vegetative cell and endospore cell. Now let's see about the difference between vegetative cells and endospores. Characteristic. Microscopic appearance of vegetative cell is non-refractile and the endospore is refractile. Calcium content in vegetative cell is low. Calcium content in endospore is high. Diplococcal acid in vegetative cell is absent. Diplococcal acid is present in endospore. Enzymatic activity is high in vegetative cells. Enzymatic activity is low in endospores. Respiration rate is high in vegetative cells and respiration rate is low or sometimes absent in endospores. Macromolecular synthesis is present in vegetative cells. Macromolecular synthesis is absent in endospores. Vegetative cells are low in heat resistant. Endospores are high in heat resistant. Radiation resistant is low in vegetative cells. Radiation resistant is high in endospores. Resistant to chemical is low in vegetative cells. Resistant to chemical is high in endospores. Lysozyme are sensitive in vegetative cell. In endospores, lysozyme is resistant. Water content in vegetative cell is high, 80 to 90%, and the water content in endospore is low, 10 to 25% in core. Small acid soluble spore protein is absent in vegetative cell. Small acid soluble spore protein is present in endospore. Thank you for your listening.